All right, well then, yes, good morning, good evening, wherever you are. I'd like to start by thanking Nathalie and all the others for the invitation, and Kelly for the kind introduction. It's a great pleasure to be able to present to you my postdoc work today on the development of cortical connectivity. So, as I'm sure most of you know, in the mammalian brain, one of the most complex structures in terms of connectivity is the cerebral cortex, as represented here in one of the historical drawings by Cajal. And we now know that there are two main classes of neurons in the cortex. The pyramidal cells that make up for about 80% of the cortical neurons and that are excitatory, and the interneurons that are the remaining 20% of cortical neurons and that are inhibitory and secrete GABA. And recent single-cell RNA sequencing studies have highlighted the wide diversity of types and subtypes of neurons within these two classes, as revealed by the many different transcriptomic profiles identified in the cerebral cortex. But then during postnatal development, all of these types and subtypes of neurons have to connect to their correct partner in a highly specific manner. And if you think about it, all the diverse set of behaviors that we observe in mammals rely on this precise assembly and fine-tuning of connectivity. So understanding the molecular mechanisms that underlie this, the formation of this connectivity remains one of the big challenges in developmental neurobiology. So in the past 5 to 10 years, a few labs have started to look at this at the level of transcription. And so this is just one example from my postdoc lab, where they've shown that 3 different types of interneurons have different transcriptional programs to regulate their wiring into cortical circuits. But then neurons have a highly complex morphology, and while transcription is restricted to the nucleus, translation or protein synthesis can take place throughout the cell body of the neuron. So in the soma, of course, but also in the dendrites, in the axons, and it was shown more recently on both sides of the synapse. And so when I joined the lab, we decided to investigate whether protein synthesis could also be differentially regulated in different cell types for their development and integration in the cortical circuits. And so in order to do this, we decided to focus on 2 types of interneurons, which are the parvalbumin interneurons, or PV cells, and the somatostatin interneurons, or SST cells. And this is because these 2 interneuron types share many developmental features. So they are born in the same place, in the medial ganglionic eminence of the subpallium. They share the same migration routes to reach their final location in the cortex, and they wire in the same cortical microcircuits. But despite these shared developmental features, the way they wire to pyramidal cells is very different, and they have very different functions in the cortex. And so that makes them interesting cell types to address this particular scientific question. And in order to modulate protein synthesis in these neurons, we decided to modulate the signaling of mTOR. So mTORC1, or mTOR complex 1, is one of these major signaling hubs in the cells that integrates external factors into downstream protein synthesis among other intracellular pathways. And in a cell, the main inhibitor of mTOR signaling is a protein called TSC2, or tuberin, that acts in a complex with TSC1 to inhibit mTOR. And so we made use of mouse genetics and the Krelok system to generate a conditional knockout of TSC2, specifically in the parvalbumin and somatostatin interneurons. So that's the LHX6 TSC2 flux mouse line. And so just that we all start on the same basis, because TSC2 is an inhibitor of mTOR, by knocking it down, we expect to have hyperactive mTOR in both types of interneurons. So this is the first thing that we checked in this mouse line on cortical sections. We looked at 2 hallmarks of the activity of mTOR that are cell size and the phosphorylation of one of the downstream effectors of mTOR called S6. And so you can see the top row are the parvalbumin interneurons, bottom is somatostatin, and you have the controls on the left, conditional heterozygotes in the middle, conditional homozygotes on the right, and the quantification. But the images really speak for themselves. You can clearly appreciate that in the homozygotes, there is a very strong increase in cell size, as well as phosphorylation of S6 for both the parvalbumin and the somatostatin interneurons. So again, this confirms our model, showing that in these mouse, mTOR is hyperactive in both the parvalbumin and the somatostatin interneurons. So then we moved on to the question that we really wanted to address, which was cortical connectivity. And the first thing that we checked were the synapses of that the pyramidal cells make onto either PV or somatostatin interneurons. And so for this, we did synaptic analysis by staining for two markers that are the presynaptic VGLUT1 and the postsynaptic PS95 that together are specific of the cortical excitatory synapses. And then we stained for either PV or SST in order to identify the postsynaptic structure of interest. So this is what the staining looks like. You have the presynapse in magenta, the presynapse in magenta, the postsynapse in cyan, and then we use a script to superimpose the two and count the number of synapses. And what we observed, so again, control in gray, conditional heads, conditional homos on the right, is that for the PV interneurons, in the absence of TSC2, we had an increased density of excitatory synapses targeting these interneurons. But then when we looked at exactly the same synapses, but this time targeting the somatostatin interneurons, we saw no change in the density of these synapses, suggesting that there might be a cell type specific impact of this mutation in terms of connectivity. And so we had this, and then I teamed up with a slice physiologist, Martijn Selten, in the lab, who recorded the excitatory postsynaptic currents received by either PV or somatostatin interneurons in these mice, and confirmed that in the mutants, the PV receive more excitation, while that doesn't change in the somatostatin interneurons. So this is what I was just saying, that this suggests that there seemed to be a cell type specific impact in terms of connectivity due to the mutation or to the lack of TSC2 in these interneurons. So then we decided to focus on the interneuron type that showed a phenotype, that's the PV interneurons, and we wanted to look at whether the output synapses of these cells were also modified. And so for this, again, we did synaptic analysis using a set of markers that are specific for the basket synapses that these parvalbumin interneurons make on the soma of pyramidal cells. So using synaptogmin 2 as a presynapse and Gefferin as a postsynapse. And again, here we saw, to my surprise anyway, no change in the density of basket synapses made from PV interneurons onto the soma of pyramidal cells. So this suggests that not only there seems to be a cell type specific impact, but also within the same cell type in the parvalbumin interneurons, there seems to be a synapse type specific impact of wiring, with the inputs that they receive being affected by the lack of TSC2, but not the output synapses that they make. So then we were curious to think that in a wild type condition, to try to identify what could be regulating TSC2 and mTOR at these synapses. And that got us into the hell that mTOR signaling is. And this is a pretty simplified already schematics. And so we decided to focus on receptors that are known to be located at this particular synapse and to be upstream of this signaling pathway. And we found one that is the receptor tyrosine kinase ERBB4. So ERBB4 is an appealing candidate for this because for two reasons. So first of all, it's expressed almost exclusively in parvalbumin interneurons in the cortex and not in somatostatin interneurons at the time of synaptogenesis. It's been shown to be located at the postsynapse of excitatory synapses targeting these cells. So it's in the, let's say, right location. And previous work has shown that ERBB4 is involved in the formation of these synapses. So if you now delete ERBB4 conditionally in parvalbumin interneurons, that leads to a reduction in the density of excitatory synapses targeting these cells. So we thought, okay, that might be a good candidate, and we came up with this hypothetical signaling pathway where, so we are now in the PV interneuron. ERBB4 is sitting transmembrane at the synapse, and when activated by its ligand, it would trigger this phosphorylation cascade downstream through AKT, TSC2, mTOR, and then the downstream effectors of mTOR. So in order to test this hypothetical signaling pathway, we turned to synaptosomes preparations. And so that's a type of fractionation assay in which you can isolate the synapses from the rest of the cortical tissue. And so we did this in the ERBB4 mouse models I just mentioned, where ERBB4 is conditionally deleted from parvalbumin interneurons. And then we did Western blots on these fractions, either the cytosolic fraction or the synaptic fraction, looking at the phosphorylation of 3 members of this pathway, TSC2 itself, as well as 2 downstream effectors of mTOR, S6 that I mentioned earlier, and 4EBP1. And when we looked first in the cytosolic fractions, so again, here you have the mutants in orange, top is phosphorylation, bottom is protein levels. We saw no obvious change in the phosphorylation of these 3 proteins or in their levels in these fractions. But when we looked in the synaptosomes, we now saw that in the absence of ERBB4, there was a reduction in the phosphorylation of TSC2 in these synaptosomes, as well as a reduction in the phosphorylation of the 2 downstream effectors of mTOR. So this suggested that the lack of ERBB4 reduces mTOR signaling at the synapse. But then in these synaptosomes, if we look at them through Western blots, we just have a mix of all of the synapses in the cortex. And so in order to check whether this was the case in our synapses of interest, instead of, we stuck to synaptosomes, but instead of doing Western blots, we plated them and we did immunostaining on them. And so for this, we used another set of synaptic markers in order to identify the synaptosomes that would represent only our synapses of interest, which are the excitatory synapses on parvalbumin interneurons. So we used PS95 as a postsynaptic marker, but we used another presynaptic marker that had been described to be specific in the cortex of the synapses that pyramidal cells make onto parvalbumin interneurons. That's neuregulin 3. And then we co-stained with phosphor-6 as a readout of mTOR signaling in these synaptosomes. And then when we looked at the ERBB4 mutants compared to control littermates, we saw a reduction in the phosphorylation of S6 in these synapses, suggesting that indeed the lack of ERBB4 leads to a reduced mTOR signaling at the excitatory synapses targeting parvalbumin interneurons. So just to recap this in the context of our signaling pathway, what we think is happening in the ERBB4 mutants is that in the absence of ERBB4 mutants, in the absence of ERBB4 from the PV interneurons, you have a reduced phosphorylation of the cascade downstream through AKT. I haven't shown you, but we have that as well. TSC2 leading to a reduced signaling of mTORC1, as we have observed by looking at the downstream effectors of mTOR. And so obviously, then we weren't going to stop there. And what we really wanted to know was that what could be affected by this downstream by this reduction in this signaling pathway at the synapse, and how that would affect protein synthesis. So in order to do this, we turned to another mouse model called the ribotag mouse. So in this mouse is a knock-in where one of the ribosomal proteins is tagged with an HA tag in a Cre-dependent manner. So that means that in your cells of interest, you can do an anti-HA pull down to pull down the ribosomes, as well as the RNA associated to the ribosomes, which is the RNA that is more likely going to be translated. And so we did this in synaptosomes that we prepared from either ERBB4 conditional knockouts or controls. And then we did RNA sequencing of these samples. And the first thing that we noticed when we looked at the transcripts that are differentially present in the ERBB4 conditional mutants or controls is that 70% of the differentially expressed transcripts are downregulated in the absence of ERBB4. So we decided to focus on these downregulated transcripts, and we analyzed what kind of proteins they would be coding for by looking specifically at synaptic proteins using a tool called SynGO, which stands for Synaptic Gene Ontology. And so it's basically gene ontology, but curated for synaptic proteins. And we found two main terms that were significantly enriched, that were postsynaptic specialization and postsynaptic membranes. And when we looked at what kind of transcripts were there, we found transcripts coding for two main families of proteins, cell adhesion molecules and empire receptor related molecules. So this was very good news for us, because when you're interested in synapse formation, cell-cell adhesion is a process of interest, and empire receptors are typically the kind of receptors that you would expect to find on the postsynapse of excitatory synapses. So then we needed to select candidates for further validation, and so for this we used a set of criteria that are shown here, including PV expression in parvalbumin, or enrichment between parvalbumin and somatostatin interneurons. And we ended up with this ranking score in pink on the right. And we selected 5 cell adhesion molecules, so calcitonin 2, neuroplastin, CADM1 that encodes for SYNCAM1, NTRK3 that encodes for TRAC-C and neuroligin 3, as well as 2 empire receptor related molecules, GRIA4 that encodes for one of the subunits of the empire receptor, and CAGNAC2 that encodes for stargazin. And so these 7 candidates, we wanted to validate them in 2 different ways, one that was more mechanistic and the other one that was more physiological, let's say. So for the more mechanistic approach, everything I've shown you so far comes from mouse models and mostly, if only, loss of function. And so I really wanted to do a gain of function experiments to see what would happen if we now activate the ERBB4 signaling pathway. And so this is what we did, again, using synaptosome preparations, this time from wild-type mice. And we treated them with the ERBB4 ligands in order to activate the pathway. And then we did Western blots for each of the 7 candidates. And we saw that following neuroregulin treatment, we had an increase in protein levels for 6 out of the 7 candidates that we were looking at. And I'm not showing it here, but we did the same experiments by pre-treating the synaptosomes with cyclohexamide, which is a drug that blocks translation. And this pre-treatment abolished the increase in protein levels that we saw for these candidates. So this again went in the direction of ERBB4 signaling at the synapse, being able to regulate the translation of these proteins. And then the second type of validation that we did had nothing to do with translation or local regulation, but we just wanted to check that these 7 proteins were involved in the formation of our synapses of interest, which are the excitatory synapses onto parvalbumin interneurons. And so for this, we designed short hairpin RNAs for each of the 7 candidates. We packaged them into CRE-dependent adeno-associated viruses that we injected into LHX screen neonates in order to generate conditional knockdowns of each of the candidates. And then at P21, we checked for the same synapses I showed you at the very beginning of this talk, so the excitatory synapses that pyramidal cells make onto PV. So this is just example images for each of the shRNA, for each of the conditional knockdowns. And we used SH-LAG-Z as a control for each of the litters. And what we observed is that in each of the conditional knockdown, we had a reduction in the density of excitatory synapses targeting the infected parvalbumin interneurons. So again, showing that these candidates are involved in the formation of the excitatory synapses onto parvalbumin interneurons. All right, so just to recap, what we have shown is a signaling pathway that is specific of a certain types of synapses, which are the excitatory synapses targeting the parvalbumin interneurons that through ERBB4, AKT, TSC2, and mTOR regulates the protein synthesis of a set of synaptic proteins that are involved in the synapse building, maturation, and probably maintenance of these particular synapses. And so in the last minutes, I would like to go back to the original questions of how neurons are wired during postnatal development, and just to think of what it means for the other synapses in the circuits. And so, I mean, I don't have the answer to this, but the question is whether a similar mechanisms would be found at other synapses, which is a different receptor and maybe a different downstream signaling, or whether there is something specific about the synapses that require this additional mechanism for their formation and maybe for their plasticity. So as I said, I don't have any answer for this, but it's interesting to think about this in the context of disorders, since these particular synapses on parvalbumin interneurons have been suggested to be a hotspot for some of the neurodevelopmental disorders. And we had a quick look at the list of genes that are found, a list of transcripts that are found downregulated in the ERBB4 mutants, and we see a significant enrichment of autism-related genes in this list of downregulated transcripts. So again, being one more building block into this hypothesis of these particular synapses being a hotspot for maladaptive network responses in some of the neurodevelopmental disorders. Right, and so I'd like to thank everyone in both labs, especially my mentors, Oscar and Beatrice, for their unfailing support throughout the project. David and Martin, who helped on this project. David helped on the validation of the candidates, and Martin did the slice physiology recordings. And then technical help from the lab and the department, Stella, Alicia, Alfonso, Forsham, Patri, Fazal, and Ian. The CRG in Barcelona, where we did the sequencing, and Genospace in Paris, who helped with the analysis of the sequencing, our funders. And then I will leave you with some art explaining why it's important to translate in order to connect. And thank you very much for your attention.